My name is Kate Donigan, and I'm a member of the personalized medicine staff within the Office of In Vitro Diagnostics at the FDA. Today, I will be discussing understanding the clinical relevance of population genetic information on the detection, characterization, and interpretation of variants, the sixth module in the UCSF-Stanford CERSI course, Regulatory Science for Next-Generation Diagnostics. In April 2016, scientists from the UCSF-Stanford CERSI joined staff from both FDA and the National Institute of Standards and Technology to publish a paper entitled A Research Roadmap for Next-Generation Sequencing Informatics. This paper lays out 9 specific research areas that will be important to answering regulatory science questions that will inform FDA regulation of next-generation sequencing diagnostic devices. As you can see in this graphic, the 9 research areas span both analytical and clinical performance of NGS tests. Today, I will be focusing on the development and use of reference data sets from different populations and how such data sets can help to drive both regulatory science and ultimately regulatory policy development. When we talk about precision medicine, we're really talking about the ability to deliver the right treatment to the right patient at the right time. But who is the right patient when it comes to next-generation sequencing? Ultimately, the success of precision medicine depends on the inclusion of diverse and underrepresented populations in both the research and the clinical settings. Some key questions to think about in the context of developing a next-generation sequencing test include both analytical and clinical considerations. First, are there any specific considerations that need to be addressed regarding the analytical detection of certain population-specific variants? For example, if an NGS test does not reliably detect a specific insertion that is only present in an underrepresented patient population, those patients may not receive the right diagnosis or treatment. On the clinical side, it is important to ask that population-specific insertion is medically relevant, keeping in mind that some variants may be disease-causing in certain populations and benign in others. A recent publication in the New England Journal of Medicine by Man Ray and colleagues highlights the importance of sequencing diverse and underrepresented populations. In this study, investigators used publicly accessible exome data to identify variants that had been considered causal in hypertrophic cardiomyopathy that are overrepresented in the general population. They found that multiple patients, all of African or unspecified ancestry, were given positive test results with variants misclassified as pathogenic, when actually they were benign. The investigators showed through simulations that inclusion of even small numbers of African Americans in the control cohorts likely would have prevented such misclassifications. The authors conclude that their results highlight the need for sequencing genomes of diverse populations, both in asymptomatic controls and in the tested patient population. They note that additional populations of different ancestry backgrounds are sequenced. They expect variant reclassifications to increase. By sequencing underrepresented patient populations and using that data in the development of next-generation sequencing tests, the promise of precision medicine, the right treatment to the right patient at the right time, will be more fully realized. The research roadmap I discussed earlier addresses the use of reference data sets that represent many different population groups and encourages the collection and characterization of high-quality samples from such groups. This characterization of samples from different populations could lead to the generation of gold standard data sets that are specific to different ethnicities and geographically defined groups. The development of population-specific reference genomes will be important for characterizing the performance of an NGS test across different populations. The availability of a suite of reference genomes from different populations will lead to meaningful test results for more patients. The research roadmap also discusses the use of bioinformatics tools to address the representation of population-specific variation in NGS tests. Regulatory science research can develop informatics tools to generate in silico genomes that account for variation that is specific to certain populations, and such genomes could then be used for benchmarking future bioinformatics pipelines. So how would the regulatory science we just discussed be applicable to the regulation of NGS-based diagnostic devices? How do we get from regulatory science to regulation? The FDA review standard for all medical devices is a reasonable assurance of both safety and effectiveness. Both terms are defined in the regulations referenced on this slide. Regarding safety, there should be a reasonable assurance that the probable benefits outweigh any probable risks. For effectiveness, there should be a reasonable assurance that a significant portion of the target population will receive clinically significant results from the device. This slide provides an overview of the different elements of FDA pre-market review in the context of in vitro diagnostic medical devices. When reviewing a diagnostic test, FDA will review both the analytical and clinical validity of the test, as well as the test's labeling. Generally, FDA considers analytical validity to be whether the test correctly detects the analyte being measured, as well as various performance metrics of the test like accuracy, precision, etc. For clinical validity, FDA wants to know if the test correctly identifies the disease or condition that it claims to identify, as well as other relevant metrics like clinical sensitivity or specificity. It is important to note that FDA can review different types of clinical data, including data from clinical studies or literature, and that randomized controlled trials are not usually required for in vitro diagnostic devices. Labeling includes any claims that a test developer wants to make regarding the analytical and clinical validity of their test. Finally, it is important to note that the assessment of both analytical and clinical validity of a test is made based on what is called the intended use of the test. We discussed FDA's review standard for safety and effectiveness earlier, and here we note that FDA applies this review standard in the context of a device's intended use. What do we mean when we talk about the intended use of a device? It comes down to three key elements. The what. What is the analyte or sample that the test will measure or detect? The who, the patient population that will ultimately be tested. And the why. What is the purpose of the test on that intended patient population? Since intended use establishes the patient population, both analytical and clinical validation studies should be performed in the intended population setting. The inclusion of underrepresented patient populations in such validation studies for NGS tests could help ensure that more patients could be included in the test's intended use. In summary, regulatory science research in the area of population genetic information will help to ensure that more people can benefit from NGS-based diagnostic tests, which will be critical on delivering the promise of precision medicine, the right treatment to the right patient at the right time.